Thank you very much for voting. Let me tell you a little bit more about SUN Bioscience, what we do and what we want to do. What I already told you is that drugs don't work for most people. And in detail, what this actually means is that any drug that is on the market, no matter what the domain, it will work for 1 out of 2 people. If you look at less understood diseases like Alzheimer and cancer, this number can go down to 1 out of 4. Now, what does this mean? It actually means that doctors don't know what drugs to prescribe to you, and the medicine that you get as a patient doesn't really work most of the time, and you still have to cope with the side effects. But if we look at the future, imagine being a patient that has Alzheimer or a colon cancer, and we can take cells from you as a patient, and really easily accessible cells, such as skin cells, and there are the protocols now in place in labs all over the world to revert these cells back into a state that is very similar to immature stem cells. And these cells can in return be used to make organoids. Organoids are nothing less than miniature organs on a laboratory dish. They have functions that have so far been only accessible inside our body. And these structures can then be used to not only model the disease, but to use for drug screenings or even make transplantable tissues because they're functional cells. And this is no longer a science fiction scenario. These structures are actually made in laboratories. We have seen the mini gut, we have seen the mini eye and the mini brain. And this is just the tip of an iceberg of what is even possible. And if we can get these structures outside of a lab and into the pharmaceutical industry, the impact will be amazing. Personalized medicine can finally come true. However, before organoids can be used for these applications, they need to become much more accessible, both in numbers and quality. And let me explain to you how they're actually made. They are made from small clusters of stem cells that are made in these plates that look like this. They're the size of an A6 format, and they're made in plastic. So you put your cells inside of one of these wells, which is round at the bottom, and they will cluster to form a cell cluster. But then they're usually transferred to a material that is more cell compatible because the plastic is simply not sufficient. What we have made is we have used the cell culture platforms that are standard. They come in many different shapes, and we have modified them with a hydrogel surface that we can restructure with wells that are based on the standard protocol, just in much smaller. And now what we can do, if the video plays, is, it doesn't play. Well, great. What we can do is we can make 500 of these clusters with just one handling step. And because it's in a hydrogel substrate, we can culture them for long-term in there. We don't need to transfer them to any other plate. And now imagine that you need 1,000 of these organoids to do a drug screening. What I haven't told you is that the protocols for deriving them from a cell cluster like this are not 100%. They're more around 10%. So of these standard plates, you would not need 10, you would need 100 of them to do your drug screening. With our platform, you can do it in one single one because you have so many repetitions. And from the video that would have played, you would have seen that actually our cell clusters form much faster, and they're highly reproducible because they're formed from one cell solution and they distribute in the miniature wells, and every single one is exactly the same. So the protocol that we have is faster, better, and way more efficient. But let me tell you a little bit about the team. We are the 2 students that have developed this platform. And it was actually the professor that you see in the center, Professor Ivan Arsenovich, who works in the Eye Hospital of Lausanne, who came to our professor, Matthias Luthoff, to ask for a platform that would allow him to derive his eye organoids faster and more efficiently. And this is what we have done. And our platform, we now use it very extensively in the 2 labs of Professor Arsenovich and Professor Luthoff, and also in the lab of Professor Olaina Veras for deriving new organoids. Recently, we have established collaborations in Denmark with Professor Grappin-Bottin, who works on pancreatic organoids, and also with Professor Jürgen Knoblich, who is actually the creator of the mini brain. So as you can see, we are spreading our platform a lot in the research world, but we would like to move beyond this, and we need your help for this. So the first thing that we are focusing on is customers. We know the customers in the stem cell field, but we know this platform has a lot of impact also in the cancer field and the tissue engineering field, and getting it to pharma We need your help with a network, or if you organize conferences or fairs where we get visibility, because we're a very young project. And once we get these customers, we need to look at the scaling of our platform. For the moment, we produce everything manually, but we're looking into ways of automating the process with a robot that would manufacture our plates, or even finding an external manufacturer and distributors that would get these plates to the people that want them. And also, we are interested in hearing about financing. As I said, we have just started 1 month ago. But we are funded on an Innovaud grant, but we are looking into investment in a year's time to maybe get the startup of 3 to 5 people running. So if you're interested in making an offer, we would love to discuss with you. Thank you for your attention.